Hello everyone, welcome back. I hope you all are doing good. So today we are going to talk about peptidoglycan structure and chemistry. This is a part 2 of bacterial cell wall. In part 1 that I previously uploaded, we discussed about introduction, historical background and overview of bacterial cell wall along with detailed understanding of gram-positive and gram-negative bacteria cell wall. Right? And today we are going to talk about peptidoglycan structure and chemistry. Peptidoglycan, which is one of the very important component of both gram-positive and gram-negative bacteria cell wall. Right? So before moving ahead, let's take lecture outline first. This topic I will be covering under these headings. Firstly, we will be talking about peptidoglycan and bacterial cell wall in brief. After that, introduction to peptidoglycan with major emphasis on its structure and chemistry. Thirdly, we will be talking about peptidoglycan subunit composition, and at last peptidoglycan cross-linking. Okay, so let's start with an introductory slide on peptidoglycan and bacterial cell wall. Okay, so peptidoglycan, we all know it is a key component of bacterial cell wall. Whether we talk about gram-positive bacteria and or gram-negative bacteria, for both types, peptidoglycan is one of the key component forming the cell wall in these type of bacteria. Okay, so now you see, as far as role and functions of peptidoglycan is concerned, then we should know that peptidoglycan is essential for maintaining osmotic stability of bacterial cell, and it also determines what? It also determines the shape of the bacterial cell. Okay, so now in brief we are going to see what are the major differences in gram positive and gram negative bacteria cell wall with respect to the presence of peptidoglycan. Okay, so when we talk about gram positive bacteria, here I have included this overview picture. You can see that cell wall in case of gram positive bacteria is made up of what? Peptidoglycan. Okay, now we are coming towards gram negative bacteria. In case of gram negative bacteria, what you can observe, that a thin layer of peptidoglycan is present when we compare it with that of gram positive bacteria. Another important point of difference you can observe, that this thin layer of peptidoglycan is also surrounded from its outside by one another membrane that we call as outer membrane, because it is present on the outside, right? And this outer membrane is absent in case of gram positive bacteria. So as far as peptidoglycan is concerned, then we should update our knowledge. The thickness of peptidoglycan layer in case of gram positive bacteria cell wall is 20 to 80 nanometer, right? On the other hand, in case of gram negative bacteria, the thickness of this peptidoglycan layer is only 2 to 7 nanometer. So what we can observe, that there is a major difference between the thickness of peptidoglycan layer in case of gram positive and gram negative bacteria. And you know, this is truly the basis of differential response shown by different type of bacteria towards gram staining procedure. We all know that when we use same steps, same gram staining procedure to stain different type of bacteria, why response is coming different? Because of the presence of such type of differences in the cell wall of gram positive and gram negative bacteria cell wall, right? In detail, I have already discussed in part 1, so you can update your knowledge from there. Here now we are coming towards our today's topic of discussion, that is peptidoglycan, an introduction first to its structure and chemistry. Okay, so now you see peptidoglycan. First, we should know that peptidoglycan is made up of what? Sugars and amino acids, right? It is actually a polymer which is made up of what? Sugars and amino acid. There is one another name of peptidoglycan, what we call as murine, right? So peptidoglycan is also known as murine. Being a polymer of sugars and amino acids, this name has been given to peptidoglycan, right? Now we are coming towards next point. Mesh-like polymer it is, which is composed of many identical subunits. With this picture, you can clearly observe mesh-like means net-like. Net-like polymer it is. Polymer means it is made up of some kind of subunits, right? So it is composed of many identical subunits. Of course, we can see here yellowish, reddish, orange, and this chain is present. These are going to be repeating here, right? And forming what? A mesh-like polymer, what we call as peptidoglycan, right? So next you see each subunit in polymer contains what? Then we should know two alternating sugars are there. Here we are going to talk in brief about subunit composition, right? So each subunit which is going to be repeated here in order to form this mesh-like polymer, it is actually containing what? Then it is containing two alternating sugar derivatives, what have been shown here in terms of this yellow coloration and reddish orange coloration. Two alternating sugar derivatives. Alternating, these are actually, right? And second point, a peptide chain of different amino acids, right? Here, this chain is actually depicting us what? It is depicting us the presence of peptide chain, which is made up of what? Amino acids, right? So peptide chain and two alternating sugar derivatives. This is actually constituting what? Peptidoglycan subunit. And these subunits are going to be repeating again and again where? In peptidoglycan structure, okay, in polymer formation. So I hope it is clear to you, the introductory part. So now we are coming towards peptidoglycan subunit composition, right? We are already clear about it. What we have observed in mesh-like polymer, two alternating sugars, right? This yellow and orange reddish color. So what we can see, two alternating sugar derivatives were there. Second, a peptide chain, right? A peptide chain which is having what? Amino acids, right? Now we should know that we have talked about it, that two alternating sugar derivatives are present, right? So what are these sugar derivatives? We need to understand about these sugar derivatives, right? Then you should know that here this picture is showing you two sugar derivatives. One sugar derivative is called as NAM and another sugar derivative is called as NAG, right? NAMate and NAG. And these amino acids, L-alanine, D-glutamic acid and so on, these are four amino acids which are going to show you the formation of a peptide chain. So this picture is actually showing us what? Peptidoglycan subunit, right? One subunit, and which are then lined up with each other to form the polymer. Let's understand in detail now about these sugar derivatives and amino acids, right? So two alternating sugar derivatives. First one we can say is NAG. NAG, what is the full form of NAG? It is N-acetyl glucosamine. It is N-acetyl glucosamine. Now we are going to talk about NAM. NAM stands for N-acetyl muramic acid. And now you should know NAM and NAG are attached to each other by means of what? Beta 1 for glycosidic bond. Beta 1 for glycosidic bond is there, which will be attaching NAM to the NAG. Okay, one sugar residue will attach to the another residue by means of this particular bond. This is very important question when it comes to MCQs part, right? Now we are coming towards another part, understanding of subunit. What is that? A peptide chain, right? Peptide chain, we all know it is actually made up of what? Peptide chain is made up of amino acids, right? And if we talk about amino acids, how many amino acids are lined up here to form peptide chain? Then we should know four amino acids. 1, 2, 3 and 4, right? Four amino acids. That's why we call it as tetrapeptide chain. A tetrapeptide chain is attached. Another important question you can say, or the point what you can learn from here, sometimes students get confused. The tetrapeptide chain is present where it is attached. It is attached to NAG sugar or NAM sugar. Then you should always keep in mind that it is attached to NAM, right? NAM through the carboxylic group of the NAM, right? Here I have also mentioned the tetrapeptide chain containing four amino acid is attached to the carboxylic group of the NAM, right? And one another point is this: what is the composition of amino acids? Many times we are knowing that tetrapeptide chain is there, four amino acids are present, but we should take care of this, that these are the four amino acids which are present: L-alanine, D-glutamic acid, mesodiamine acid and D-alanine, right? I have also mentioned here. Next point you should know, in case of many gram-positive bacteria, there is an exception that in many gram-positive bacteria, in place of mesodiamine acid, one another amino acid is present, right? What we call as L-lysine, right? L-lysine usually replaces what? Mesodiamine acid in case of gram-positive bacteria. So here I have clearly written many gram-positive bacteria replace mesodiamine acid with another diamino acid, usually L-lysine, right? Now one more thing you need to make it clear, that why L-lysine is going to replace mesodiamine acid, why not D-glutamic acid or L-alanine? Then you should update your knowledge that L-lysine and mesodiamine acid, they both are diamino acid. Diamino acid means they both are having two amino groups, right? One amino group in this amino acid is present to form peptide bond with another amino acid, adjacent amino acid, and the side chain amino group present in these amino acids are usually involved in forming crosslinks, right? Like this. In this picture you can see. So here what is there? Suppose one chain of peptidoglycan is there. On the other hand, another chain is there. How these chains will be attached to each other? These chains will be attached to each other by means of cross-linking, right? That I'll be discussing after this slide. So these are attached to each other by means of this cross-linking. And during cross-link formation, what is required, some amino acids should be like this, that they should have an extra amino group, right? So that amino group can form cross-linking with the help of formation of peptide bond with terminal amino acid of another chain. So in this way, chains are linked up. I'll be telling you further or making it you clear about it. But here you need to know about it that L-lysine is going to replace what? Mesodiamino-pymalic acid. Both are diamino acids, and diamino acids means both two amino groups they are having. Why these two amino groups are required? One for forming bond with the adjacent amino acid, and another for making cross links, so that different chains of peptidoglycan can be attached to each other, right? One another point is there which is very important from competitive examination point of view. So what you can see here, in this tetrapeptide chain, three of these amino acids are what? D amino acids, right? So what is the advantage these D amino acids are actually giving to bacteria? So you should make your understanding clear that what actually happens, that most of the peptidases actually target L amino acids, right? Peptidases which attack proteins, they actually target L amino acids, and when majority of the D amino acids are present in case of bacterial cell wall, then they become resistant to attack by peptidases, and they will not, the cell wall of bacteria will not be undergoing any kind of disintegration, right, or will not be damaged after the attack of the peptidases. So the presence of D amino acid is actually an added advantage for bacteria, right? So now we are coming towards next slide. So here you can just see, if you are interested to know about the structures, then this slide will be beneficial for you, otherwise you can skip it. So what I have tried to show you here, sometimes in order to attempt our answers, what we need to also draw the structure. So here I just would like to give you major highlights, main highlights. So what is that? NAM and NAG, these are attached with each other through what? They are attached with beta 1,4 glycosidic bond, right? And another thing you should know, that how NAM is out of NAM and NAG is attached to tetrapeptide chain, then NAM is attached with tetrapeptide chain. NAG is not having any tetrapeptide chain. If NAM is attached with tetrapeptide chain, through which group of it is attached to the tetrapeptide chain of amino acids, then you should know NAM is attached with tetrapeptide chain mainly through its carboxylic acid group, right? And subsequently what will be there? Four amino acids are attached to each other by means of which bond? By means of peptide bond, right? Peptide bond is actually the bond which is formed between amino group of one amino acid, right, and carboxylic acid group of another amino acid. So secondly, if you are interested to draw the identical subunits or the peptidoglycan subunit for gram-positive bacteria mainly, then you can also use the structure of L-lysine here in place of mesodiamino-pymalic acid. Okay, and here what is shown, that this terminal D-alanine is actually attached to what? It is actually attached like this to another chain of peptidoglycan. Okay, so these are the structural informations. And one more point here, peptidoglycan subunits being rich in D amino acids also make peptidoglycan a very important source of D amino acids in nature, right? So this point can also be of use to you. That's why I thought of including. Now we are coming towards peptidoglycan crosslinks. So what are peptidoglycan crosslinks? To make a strong mesh-like polymer, chains of linked peptidoglycan subunits must be joined to each other by means of crosslinks between the peptides. Okay, so already we have seen that picture what I have initially shown you, that peptidoglycan is what? A strong net-like polymer, right? So how this type of polymer will be resulted? Then we should know that it will result only when different chains of peptidoglycan having identical subunits are in some way joined or attached to each other, right? And this joining or attachment of different chains having identical subunits of peptidoglycan is called as what? Cross-linking, right? Cross-linking is what? Cross-linking is joining of chains of linked peptidoglycan subunits. If simply I define what it is, joining of chains of linked peptidoglycan subunits is called as what? Cross-linking, right? So, and cross-linking occurs between what? Then you should know whether sugars and sugars are attached to each other. Cross-linking is between sugar and sugar, or sugar and tetrapeptide, or tetrapeptide and tetrapeptide. Then you should be clear about it that cross-linking occurs between peptides, okay, between peptide chains. It is not between sugars. Okay, this picture will further make you clear about it. So what I have tried to show you here, these are alternating NAG and NAM subunits which have been shown here also, and how they are attached to each other. This small line is indicating the presence of tetrapeptide chain on the NAM residue. What it is going to show you, you can see here that NAM is having what? Tetrapeptide chain, and this is another chain, it is also having NAM with tetrapeptide chain. These tetrapeptide chains are linked to each other by means of what? Cross-linking, right? Their joining is called as what? Cross-linking, and this type of some bond is there, and that bond formation is called as cross-link formation, right? Cross-linking will be there. So what are the types of cross-linking, how cross-linking occurs, that we need to see further. Okay, so now we are coming towards types of cross-links in case of peptidoglycan. Then you should know this is picture A, right? And here this picture is showing us what? It is going to show us peptidoglycan cross-linking for many gram-negative bacteria. So in case of many gram-negative bacteria, how different chains of peptidoglycan are cross-linked with each other, then you should know here terminal D-alanine, right? Often the carboxyl group of terminal D-alanine is connected to the amino group. I have already told you diaminopymalic acid is what? It is usually present in case of gram-negative bacteria, and it is a diamino acid. One amino acid form peptide bond with adjacent amino acid, and another amino acid which is present in its side chain, that usually form bond with what? Terminal D-alanine of another chain. This we call as cross-linking, right? So same what I have explained you, it is also I have written, often the carboxyl group of terminal D-alanine is connected directly to the amino group of diaminopymalic acid of many gram-negative bacteria. This happens in case of E. coli. So in this way, two chains are attached to each other, and this we define as what? Cross-linking. Now we are going towards gram-positive bacteria. In case of gram-positive bacteria, I already told you that diaminopymalic acid is most of the time replaced with L-lysine, right? So here, terminal D-alanine of one chain, identical subunit chain of peptidoglycan is attached or is joined in order to form cross-link to another chain of identical subunits of peptidoglycan by means of an interbridge, right? What we call as peptide interbridge. So what is that interbridge? Here, commonly found in case of gram positive bacteria, right? As in most gram negative bacteria, cell wall peptidoglycan lacks the peptide interbridge. What is that peptide interbridge? It is again made up of a peptide chain which is having 5 amino acids and same kind of amino acid, what we call as glycine. So five glycine residues are there forming a peptide chain, and that act as an interbridge for connecting, for cross-linking D-alanine with L-lysine, right? So I hope this will be clear to you, and this peptide interbridge is absent in case of gram-negative bacteria, and it is present in case of gram-positive bacteria. So this type of cross-links, we can say two types of cross-links are there to attach identical subunits of peptidoglycan chains in order to form a mesh-like polymer, what we call as peptidoglycan, right? So now you see this cross-linking is very important to bacterial cell. Okay, without this cross-linking, what will happen? Peptidoglycan layer will not be intact, wall will not be strengthened, right? So there are some antibacterial agents which specially target these cross-link disruption, right? Like you see, we all know about lysozyme. Lysozyme is well known for its antibacterial properties, and do you know that this lysozyme actually act where? It actually having a target site beta-1,4 glycosidic bond. It breaks down beta-1,4 glycosidic bond between NAM and NAG, and in this way the cell wall of bacteria will be disintegrated and bacteria undergo killing, right? So in this is actually the mode of action of lysozyme, what you can clearly understand through this picture. And one another point I would like to tell you about penicillin. Penicillin, we all know these are wonder drug, right? And discovered by Alexander Fleming. So penicillin, what is the mode of action of penicillin? So again, you need to know that penicillin acts on where? It does not act on breakdown of this bond. It actually acts on what? Disruption or breakdown of this cross-linking, right? By means of what? This cross-link formation is actually catalyzed by an enzyme what is called as transpeptidases. Transpeptidases are the enzymes which catalyze this reaction, that how D-alanine will get attached to L-lysine by means of this peptide interbridge, right? And this cross-linking is actually targeted or attacked by penicillin. When this cross-link formation will not be there by means of inactivation of that transpeptidase enzyme, what will be there? bacterial cell wall will not be intact. Peptidoglycan layer will not be intact and cell will undergo lysis, right? So now we are coming towards importance of cross-linking. What is the importance of cross-linking? So first we should know it results in an enormous peptidoglycan sac that is dense and interconnected network mesh-like polymer. It will only be resulted when cross-linking will be there. Second point, we should know that these sacs of peptidoglycan have been isolated from gram-positive bacteria and are strong enough to retain their shape integrity, right? What has been found that to retain their shape and integrity, they are very much responsible, right, because of their strong nature. But in addition to this, it has also been found that they are relatively porous, elastic, and somewhat stretchable, right? So these are some of the important points that why this cross-linking is important in case of peptidoglycan. I hope this information will be useful to you. Thank you so much. Stay connected.